So we would have left off just introducing the metabolism of muscle here. And so we began this PowerPoint by speaking about the three mechanisms of muscle to generate ATP to sustain that contraction. We talked about the creatine phosphate system. We said that that was a transient mechanism, just enough to warm up the muscle, just enough to give ATP while anaerobic glycolysis and or oxidative phosphorylation actually was able to generate ATP to take over sustaining that muscle contraction. We then talked about the other two mechanisms, which were that anaerobic glycolysis and that oxidative phosphorylation. Anaerobic glycolysis, we said, could happen absent oxygen. We'll see how that will play a role in what types of activity can be sustained with anaerobic glycolysis. But basically, we don't need oxygen to do this, but it's going to provide a little bit less ATP. It's not as efficient a source as oxidative phosphorylation. This is something we talked about when we looked at cellular respiration. So it does generate ATP, not as much, but the caveat is that we can do it absent oxygen. Which may not always be present when we're doing activity, physical activity with muscle. The other mechanism is oxidative phosphorylation, much more efficient, right? Much more ATP made. But again, the drawback here is that we need oxygen. Oxygen must be present. This happens in the mitochondria. This is that electron transport chain mechanism of ATP production. And so this is going to be more sustainable, right, for long-term physical activity. But the understanding is that there must be oxygen to drive oxidative phosphorylation. Okay, now on that basis, we can talk about how muscles use energy or change their energy source with different intensity of exercise. And this will also feed back into that understanding of whether or not oxygen is available. So the first one is what happens, how do muscles generate ATP with light to moderate intensity? We're thinking walking here, a brisk walk, walking around shopping center, maybe some jogging, light lifting, maybe cleaning. We generally start out with using creatine phosphate, again, just getting things warmed up, that transient mechanism. Then we tap into glycogen stores. So any glycogen that is in the body, we can start tapping into that and breaking that down. Up to 30 minutes, we can also tap into what's already in the blood. So let's say it's you've had a meal recently that has glucose, you have glucose actively circulating in the blood, you've got fatty acids actively circulating in the blood. Up to 30 minutes, we can tap into those stores that are circulating in the blood, right? Beyond that period, beyond that time frame, after we've exhausted glycogen stores, after we've exhausted the circulating reservoirs, so fatty acids or glucose that's in the blood, 30 minutes or so, we then need to tap into oxidative phosphorylation. And so oxygen supply must be kept adequate. That's why we can do this with light to moderate intensity exercise, okay? So if you're walking or jogging or cleaning, you can maintain your rate of ventilation. You can even increase your ventilation to continue bringing in adequate amount of oxygen to drive oxidative phosphorylation. And as you can see over here, oxidative phosphorylation is going to take over and sustain that type of light to moderate intensity activity. We also want to think about increasing your heart rate and your contraction, and basically just allowing for the circulation of that oxygen. So bringing it in via ventilation is not enough. We want to increase the rate of the heart to kind of pump that oxygen around the body via the circulatory system and the blood. And then we want to think about dilating vessels to muscles as well. So again, delivering that oxygen from the lungs where we breathe it in, from the heart where it's being pumped out, dilating our vessels so that it can actively be transported out to the muscles, again, sustaining that oxidative phosphorylation by delivering that oxygen directly to the muscle tissue. Now here we're going to insert transiently some GLUT4 receptor or GLUT4 transporter, excuse me. These were the transporters that we can insert into the membrane of our cell as a way to help us uptake and use that glucose. And so again, in order to sustain this, we're going to transiently increase those GLUT4 receptors to uptake any glucose that's circulating in the blood within that 30-minute window. All right, so this is how our muscles generate the ATP that is needed when we're doing lighter, moderate intensity activities. All righty, and oxidative phosphorylation is the mainstay of that. Let's contrast that to how our muscles generate ATP with heavy intensity exercise. So heavy lifting weights at the gym, running, sprinting, any type of vigorous or strenuous activity, right? Sports and athletics and karate and any type of more heavier intensity, more involved exercise. This is where we're going to shift over to fully relying on anaerobic glycolysis, okay? So what we see here is we start out as usual with creatine phosphate, getting things warmed up, but then oxidative phosphorylation is happening in the background, but it's not enough. We're not able to maintain a comfortable rate of ventilation. If you're running a marathon or if you're lifting heavy weights, your ventilation is just not adequate to sustain oxidative phosphorylation to supply the muscles with the amount of ATP that they need for this heavier intensity exercise. And so we're going to shift our focus from oxidative phosphorylation to anaerobic glycolysis. Anaerobic glycolysis will supply the bulk of the ATP that we need, but it will do so very quickly, and it can do so absent oxygen. That's the most important thing here. All right. Now, the drawback here, right, both of these mechanisms have their drawbacks. The drawback of anaerobic glycolysis is that we produce lactate or lactic acid as a byproduct. So because we're not driving oxidative phosphorylation, because we don't have oxygen, there's a backup of pyruvate. Remember, pyruvate is what happens at the end of glycolysis. Pyruvate should be shuttled off towards the Krebs cycle, the linking step, the Krebs cycle, and eventually into oxidative phosphorylation. But if oxidative phosphorylation is not being driven, it's not happening because oxygen is no longer being supplied adequately, we have a backup of all of those pathways, including backing right up to pyruvate. And then pyruvate can get shuttled off to lactic acid as a temporary way of shuttling that pyruvate because it's not being used by the other pathways, all right? And so we make this lactic acid. This is what gives us the burn in muscle that we see when we're lifting heavy weights, again, running, sprinting, jogging. That burn that's felt in the muscle is a result of the production of lactate, which is felt as an acidic lactic acid in the muscles. Okay. Another drawback here with anaerobic glycolysis is that we just don't supply enough ATP. It's a small amount of ATP, but it can be done quickly. It can be done a lot more quickly, as we can see that this starts out raising a lot more faster than oxidative phosphorylation. So while it is a less amount of ATP, while it starts more quickly, we have that lactic acid as a price to pay. All right. What we're going to speak about now are the fiber types in muscle, okay? How do we classify skeletal muscle? So all skeletal muscles are not created equally, and they differ based upon two things. They differ based upon their velocity of contraction, how fast they can contract versus how slow they contract. So that's kind of one classification of muscle. The other one is its primary source of ATP production. Does it primarily rely on oxidative phosphorylation, or does it primarily rely on anaerobic glycolysis? And as we will see, that basis, its primary mechanism of generating ATP, is going to create a lot of physical features that differ between muscle types, okay? All right, so let's talk about the first name of the muscle. So essentially, if we think about, let's go back here. If we think about these two bases of classification, the velocity of contraction can be thought to be the first name of the muscle. Just like you have a first name and a last name, a muscle has two names as well. The first name is its velocity of contraction. It's fast or slow. The second name is its primary source of ATP production, whether it's oxidative or glycolytic. So we can have fast oxidative fibers, we can have fast glycolytic fibers, we can have slow oxidative fibers, okay, based upon that classification. So let's talk more about that first basis for classification, which is the speed of contraction. So we can have fast twitch fibers and slow twitch fibers. And what differs here is the rate of myosin ATPase activity. So we talked about the myosin head having ATP bind on there, that ATP then being hydrolyzed to change the state of that myosin head to make it want to interact with actin. The rate at which that can happen, the rate at which that hydrolysis of ATP can happen is going to determine the rate of cross-bridge cycling. So if the myosin can hydrolyze ATP very quickly, form a cross-bridge cycle very quickly, release that cross-bridge cycle very quickly, we're going to call it a fast twitch fiber because it has a faster rate of cross-bridge cycling, right? It's going to be able to contract more quickly because of that ATPase activity. So again, on that basis, we have fast fibers. These have myosin, which has a fast ATPase activity. It can make and break those cross-bridge cycles really fast. We can then compare that or contrast that to slow fibers. They have a slow rate of, or the myosin heads rather, have a slow rate of ATPase activity. That means they're not going to make and break those cross-bridge cycles as quickly as our fast fibers. Going on further here to compare the fast and slow fibers. So fast fibers can contract 2 to 3 times more rapidly than slow fibers. And so that is to say that a slow fiber contraction would last probably 10 times that of a fast fiber contraction. So if I were to think about the shortening of a slow fiber, it just takes so long, right? It takes 10 times longer that a fast fiber has already done 3, 4 times the amount of contractions because the slow fiber contraction just took so long, or the slow fiber took so long to complete a single transaction, okay? Another factor that differs here between fast and slow fibers is the rate of relaxation. That is the rate of the calcium ATPase pump as well. So just going back here, taking us back to our initial muscle phys conversation, taking us back to excitation contraction coupling, we said that there was a connection between the rate of muscle relaxation and the pumping of calcium back into the sarcoplasmic reticulum, right? So in order for us to put that muscle back into its relaxed state, we're going to need to take that calcium off in order to cover up those binding sites again. And we're going to need to push that calcium from the sarcoplasm back into the sarcoplasmic reticulum where it is being stored. And calcium ATPase is the pump that helps to pump that calcium from the sarcoplasm back into the sarcoplasmic reticulum. And so if we can pump the calcium back into the sarcoplasmic reticulum faster, we can relax the muscle faster, right? We can clear that calcium out faster, the muscle can relax faster, and guess what? If muscle can relax faster, it can initiate a new contraction faster as well. So the difference between fast fibers and slow fibers is not only found in their rate of myosin ATPase activity, that is their rate of cross-bridge cycling, but also there's a faster rate of calcium ATPase activity, which is their rate of relaxation as well. So they can contract faster, they can also relax faster, and they can move in general much faster than slow fibers. Okay, here's just an illustration of that, just really kind of showing you the difference between the time it takes for a slow fiber to get to its maximum percent of tension production or force generation versus the time that it takes for a slow fiber to do that. So here are an example of 3 different types of fibers. We have the extraocular muscles. They can get to their maximum contraction in about 7 to 8 milliseconds. Comparing that to the gastrocnemius muscle, which is the calf muscle, they can get to their maximum contraction in about 40 milliseconds, so a longer period of time. And then contrasting that to a much slower fiber, the soleus muscle. The soleus muscle can get to its maximum contraction in about 90 milliseconds, okay? And so we can see that it takes a long time for this this muscle to get to its peak of contraction, much less complete its contraction in its entirety. And so within that time, we can have an extraocular muscle complete 3, 4, 5 contractions fully in the time it takes a soleus to complete a single contraction. Okay. What I also want to point out is we can make a connection between the rate of the contraction and relaxation of these 3 muscles with the physical location, right? The anatomy of these muscles and the type of activity that these muscles are most likely doing. So our extraocular muscles are the muscles of our eyes. These are the muscles that help to move our eyes in terms of tracking and maintaining vision. And so they're not doing a whole lot of strenuous activity. They're not doing a whole lot of physical movement, or they're not doing a lot of strength movement, that is. They're more just kind of moving the eyes around. And so it would make sense that these muscles are faster muscles. They're more reactive. They have a faster myosin ATPase rate, faster calcium ATPase rate, and that would make sense for muscles of the eye. Contrasting that to the soleus muscle. The soleus muscle is one of the deeper calf muscles. We would have talked about this in lab this week. It is a muscle for posture and support. So this muscle can maintain slow, long, steady contractions because it has to maintain the weight of the body for long periods of time. So if you're standing for an hour, this is the muscle that's going to kick in and take over and maintain those longer, slower contractions that are important for posture and for support. Okay. If you're walking as well, walking is a part of that, the use of this muscle.